Hello students, welcome to readmedprepacademy channel. Today in Principles and Processes of Biotechnology Part 4, we are going to talk about transgenic plants, otherwise called genetically modified crops, biofarming, bioremediation, biofuel, bioprospecting and biopiracy. Let us talk about the herbicide tolerant plants to glyphosate. Weeds are a constant problem in the crop fields. Weeds not only compete with the crops for sunlight, water, nutrients and space, but also acts as a carrier for insects and diseases. If left uncontrolled, weeds can reduce the crop yield significantly. Transgenic plants contain a novel DNA introduced into the genome. Produced by Monsanto USA company under the trade name Roundup, kills plants by blocking the 5-enopyruvate shikimate 3-phosphate synthetase, otherwise shortly formed EPSPS enzyme, an enzyme involved in the biosynthesis of several aromatic amino acids, vitamins, many secondary plant metabolites. What is the effect of glyphosate on the plants? When the glyphosate is applied to the plant or sprayed on the crops to remove the weeds, they persist in the environment, glyphosate residues, and they form metabolites like the AMPA, and these are again taken up by the crops. Sometimes they interact with the nutrient availability, like they cause chelation of the micronutrients. Competitive adsorption with phosphates occurs, and they also cause effects on the crop's health. What is the defect that is caused? First, let us start the metabolism. Shikimic acid in the presence of shikimate kinase forms shikimic acid 3-phosphate. Shikimic acid 3-phosphate in the presence of PEP or EPSP synthase forms 5-enol pyruvyl shikimic acid 3-phosphate, which in the presence of corismate synthase forms corismic acid. Corismic acid results in the synthesis of phenylalanine and tyrosine, or it helps in the formation of anthralinic acid, which results in the formation of tryptophan. Glyphosate blocks the conversion of shikimic acid 3-phosphate in the presence of EPSP synthase to 5-enol pyruvyl shikimic acid 3-phosphate. That is the where it works. So there are several ways by which crops can be modified to be glyphosate tolerant. One strategy is to incorporate a soil bacterium gene that produces a glyphosate tolerant form of EPSPS. Another way is to incorporate a different soil bacterium gene that produces a glyphosate degrading enzyme. How the glyphosate resistant crops work? The target plasmid is taken and the glyphosate insensitivity EPSPS from a bacteria is taken and is incorporated into the plasmid, and a chloroplast targeting sequence is present and a CAMV promoter is present. This is agrobacterium mediated DNA transfer occurs into the tobacco plant, and this EPSPS from the bacteria is taken up by the plant cell into the cytoplasm or into the chloroplast. When the glyphosate is sprayed in a wild type, the plant withers or dies like the weeds. In a transgenic plant which is having the agrobacterium mediated DNA gene, the endogenous plant EPSPS is inhibited by the herbicide, whereas the bacterial EPSPS is still active. Hence the plant survives. What are the advantages of herbicide tolerant crops? Weed control improves the higher crop yields, reduces spray of herbicide, reduces the competition between the crop plant and the weed, use of low toxicity compounds which do not remain active in the soil, the ability to conserve the soil structure and microbes. Now let us talk about the herbicide tolerant Basta. The trade name Basta refers to a non-selective herbicide containing the chemical compound phosphenothricin. Basta herbicide tolerant gene PPT, otherwise L-phosphothricin, was isolated from Medicago sativa plant. It inhibits the enzyme glutamine synthase, which is involved in ammonia assimilation. The PPT gene was introduced into the tobacco and the transgenic tobacco produced was resistant to PPT. Similar enzyme was also isolated from Streptomyces hygroscopicus with BAR gene encodes for PAT, phosphinothresin acetyltransferase, and was introduced into the crop plants like potato and sugar beet, and transgenic crops have been developed. Now let us talk about the insect resistance BT crops like BT cotton. It is a genetically modified organism or genetically modified pest resistant plant cotton variety which produces an insecticide activity to bollworm. Strains of the bacterium Bacillus thuringiensis produce over 200 different BT toxins, each harmful. Here you can see in this image the Bacillus thuringiensis, which has an endospore on one place and a nucleoid having the DNA and the ribosomes, the cell membrane outside which is the cell wall. In it you can see a BT toxin crystal. Most BT toxins are insecticidal to the larvae of moths and butterflies, beetles, cotton bollworms and gadflies, but are harmless to other forms of life. The genes are encoded for toxic crystals in Cry group of endotoxin. When insects attack and eat the cotton plant, the Cry toxins are dissolved in the insect's stomach. The epithelial membranes of the gut block certain vital nutrients. Thereby, sufficient regulation of potassium ions are lost in the insects and results in the death of epithelial cells in the intestine membrane, which leads to the death of a larvae. Here you can see the transgenic plant having the crystals and spores of BT toxin and are ingested by the insect larvae. And the toxins are activated to active form by the gut enzymes of the worms. And the activated toxin binds to the receptor, subsequently inserts into the membrane and cause leakage of the ions and small molecules. The midgut membrane damage occurs, leading to starvation or septicemia of the worm and the death of the worm. What are the advantages of BT cotton? Yield of cotton is increased due to effective control of bollworms. Reduction in insecticide use in the cultivation of BT cotton. Potential reduction in the cost of cultivation. What are the disadvantages? Cost of BT cotton seed is high. Effectiveness up to 120 days after that efficiency is reduced. Ineffective against sucking pests like jassids, aphids and whitefly. Affects pollinating insects and thus yield. Now what is BT brinjal? The BT brinjal is another transgenic plant created by inserting a crystal protein gene Cry1AC from the soil bacterium Bacillus thuringiensis into the genome of the various brinjal cultivars. The insertion of the gene into the brinjal along with other genetic elements such as promoters, terminators and antibiotic resistance marker gene into the brinjal plant is accomplished using agrobacterium mediated genetic transformation. The BT brinjal has been developed to give resistance against Lepidopteran insects, in particular the brinjal fruit and shoot borer, Lucinoides orbonalis. Here you can see the insect Lucinoides orbonalis. 50 to 90% of the damage is caused by the fruit and the shoot borers. On the left side you can see how the shoot is affected and damaged by the borer, and on the right side you can see the holes caused by the fruit borer and the damage to the fruit. Now what is this Dhara mustard hybrid DMH? The DMH11 is a transgenic mustard developed by a team of scientists at the Center for Genetic Manipulation of Crop Plants, Delhi University, under government sponsored project. It is genetically modified variety of herbicide tolerant mustard. It was created by using Barnase or bar star technology for genetic modification by adding genes from soil bacterium that makes mustard a self-pollinating plant. DMH11 contains 3 genes, with bar gene, Barnase and bar star sourced from soil bacterium. The bar gene had made plant resistant to herbicide named Basta. You can see on the left side the Dhara mustard hybrid, and on the right side it is a transgenic crop developed by the Center for Genetic Manipulation of Crop Plants at Delhi University. Now what is the virus resistance crops? Many plants are affected by virus attack, resulting in serious loss in yield and even death. So biotechnological intervention is used to introduce viral resistant genes into the host plant so that they can resist the attack by the virus. This is by introducing genes that produce resistant enzymes which can deactivate the viral DNA. Here you can see the development of the virus resistant crops, RNAi mediated, to construct to generate siRNA and miRNA from the transgene, which helps in the transcription of HpRNA and dsRNA. With the help of DISA and RISC, the target specific siRNA or miRNA is formed. It has a sequence specific degradation of viral RNA conferring resistance. On the right side you can see the protein product mediated development of virus resistant crops. The virus transgene or R gene from the host plant undergoes transcription and that results in the formation of messenger RNA. And the translation results in the formation of a protein, and the specific protein plants confer viral resistance. Now, what is this flavor savour tomato? Agrobacterium mediated genetic engineering technique was followed by to produce flavor savour tomato, that is the retaining the natural color and flavor of the tomato. Through genetic engineering, the ripening process of tomato is slowed down and thus prevent it from softening and to increase the shelf life. The tomato was made more resistant to rotting by agrobacterium mediated gene transfer mechanism, introducing an antisense gene which interferes with the production of the enzyme polygalacturonase, which help in delaying the ripening process of tomato during long storage and transportation. You can see in this image the upper part, the normal tomato having the normal messenger RNA results in the formation of protein by translation, and this causes natural rotting due to the PG. In a transgenic tomato, the PG gene is introduced into the nucleic acid and a sense messenger RNA is formed. And the antisense messenger RNA is formed and a synthetic antisense gene is present in the RNA. And this helps in the formation of complementary messenger RNA. This prevents the ripening and the rotting of the tomatoes for about 3 weeks. Now, let us move on to the golden rice biofortification. Golden rice is a variety of Oryza sativa called rice produced through genetic engineering of biosynthesized beta carotene, a precursor of vitamin A, in the edible parts of rice developed by Ingo Potricus and his group. The aim is to produce a fortified food to be grown and consumed in areas with a shortage of dietary vitamin A. Golden rice differs from its parental strain by addition of 3 beta-carotene biosynthesis genes, namely PSY, photoene synthetase, from daffodil plant Narcissus pseudonarcissus, and CRT1 gene from the soil bacterium Erwinia aureodorora, and LYC gene or lycopene cyclase gene from the wild type rice endosperm. How this is done, you can see the pathway. Beta-carotene pathway, how the genes are added. IPP, isopentenyl diphosphate, forms geranyl diphosphate, and the daffodil gene is added. This, in the presence of phytoene synthase, forms phytoene, and in the presence of the single bacterial gene and in the presence of phytoene desaturase, the phytoene forms the lycopene. Lycopene, again in the presence of the daffodil gene, in the presence of lycopene beta cyclase, forms beta carotene, which is a vitamin A precursor. The vitamin A pathway is complete and functional in the golden rice. The endosperm of normal rice does not contain beta carotene. So golden rice has been genetically altered so that the endosperm now accumulates the beta carotene. This has been done using recombinant DNA technology. Golden rice can control childhood blindness like xerophthalmia. So what are the benefits of the genetically modified foods? High yield without pests, 70% reduction of the pesticide usage, reduce soil pollution problem, conserve microbial population in soil. What are the risks believed to occur? Affect the liver, kidney function and cancer, hormonal imbalance and physical disorder, anaphylactic shock or sudden hypersensitivity reaction and allergies, adverse effect in the immune system because of the bacterial proteins, loss of viability of seed seen in terminator seed technology of GM crops. Or what are the benefits and risks of genetically modified organisms? Nutritional value of foods could be improved. Example, by introducing proteins, vitamins or vaccines. New traits could cause adverse health reactions. Example, new proteins may cause allergic responses. Crops can be produced that lack known allergens. Removal of traits could have unknown effects. Crops can grow in arid conditions for better yield. Example, by introducing drought-resistant genes. The risk, crops may limit biodiversity of local environment, increase competition with native species. The genetically modified crops can produce herbicides to kill pests, whereas the cross-pollination could lead to superweeds is one of the risks. Improve food supply, agriculture in poor countries. So genetically modified crops can be engineered for improved yields, whereas the patents restrict farmers from accessing genetically modified seeds. Biotech companies hold monopolies over the crop usage. Genetically modified crops may have longer shelf lives or less spoilage, whereas the risk, foods with genetically modified components may not be labeled. Reduces economic costs and carbon footprint, less need for land clearing and pesticide usage, whereas risks, different governments may have conflicting regulatory standards concerning the safe usage. Now, what is polyhydroxybutyrate? PHB. Synthetic polymers are non-degradable and pollute the soil, and when burnt add dioxin in the environment, which may cause cancer. So efforts were taken to provide an alternative eco-friendly biopolymers like polyhydroxyalkanoates, PHAs, and polyhydroxybutyrate, PHB, are group of degradable biopolymers which have several medical applications such as drug delivery, scaffold and heart valves. PHAs are biological macromolecules and thermoplastics which are biodegradable and biocompatible. Several microorganisms have been utilized to produce different types of PHAs, including gram-positive like Bacillus megaterium, Bacillus subtilis, and Corynebacterium glutamicum. Gram-negative bacteria like group of Pseudomonas species and alkali genes eutropus. Here you can see Delphtia acidovorans, which is a micro-producing PHA, Bacillus megaterium, Cupriavidus necator. What is the structure of PHA? It has a surface layer of granule associated proteins. Inside is an intragranular water. Inside that is an amorphous PHA. How the commercial production of polyhydroxybutyrate is done from bacteria? Organic solvent is used to release the PHB from the cells, and centrifugation is done, and it is put in an evaporator and a palletizer and a bagging machine. Finally, the PHB powder is obtained. Now, what is polylactic acid or PLA? Polylactic acid or polylactide, PLA, is a biodegradable and bioactive thermoplastic. It is an aliphatic polyester derived from renewable sources such as corn starch, cassava root, chips or starch or sugarcane. For the production of PLA, two main monomers are used, lactic acid and the cyclic diester lactide. The most common route is the ring opening polymerization of lactide with metal catalyst like tin octoate in solution. The metal catalyzed reaction results in equal amount of D and polylactic acid. How is PLA manufactured? First, the sugar beets of corn are used to extract the dextrose, and the fermentation is done where the reduction of lactic acid occurs. This lactic acid is made into a pre-polymer, and then a lactate formation is done. The lactate formation is again put in a distillator to cause distillation. The distillation can also result in the formation of lactic acid again, or it can be converted into mesolactide and low D lactide, which undergoes polymerization by coordination and insertion propagation, and PLA is formed in the form of a powder. The green fluorescent protein is a protein containing 238 amino acid residues of 26.9 kDa that exhibits bright green fluorescence when exposed to blue to ultraviolet range 395 nanometers. GFP refers to the protein first isolated from the jellyfish Aquaria victoria. GFP is an excellent tool in biology due to its ability to form internal chromophore without requiring any accessory cofactors, gene products, enzymes or substrates other than molecular oxygen. In cell and molecular biology, the GFP gene is frequently used as a reporter of expression. It has been used in the modified forms to make biosensors. Here you can see the production. The green fluorescent protein is isolated from Aquaria victoria, which is a jellyfish, and a gene splicing technique is done, resulting in alteration of the codon, and this is introduced into Arabidopsis thaliana plant, and the GFP is expressed in Arabiana thaliana plant, and the GFP from Aquavira results in the formation of GFP, and the GFP is produced in Arabidopsis thaliana. Biofarming. Biofarming is also known as molecular farming, is the production and the usage of transgenic plants genetically engineered to produce pharmaceutical substances for use of human beings. This is also called molecular farming or genetic farming. These plants are different from medical plants which are naturally available. How biofarming is done? The candidate protein which is responsible is present in a structural gene. This structural gene is inserted into a plant expression vector and is introduced into a plant, resulting in the formation of transgenic plants, and a commercial production of the plantation is done and the harvest of the plant is done, and the required material or the drug is extracted and purified and is sent to the pharmaceuticals. The use of plant systems as bioreactors is gaining more significance in modern biotechnology. Many pharmaceutical substances can be produced using transgenic plants, example golden rice. What are some of the products of biofarming? Avidin, traditionally isolated from chicken egg whites, used in medical diagnostics. Beta glucuronidase, which is traditionally isolated from bacterial sources, E. coli, used as a visual marker in research labs. Trypsin, traditionally isolated from bovine pancreas. It has variety of applications like biopharmaceutical processing. It's a first large-scale transgenic plant product. Now, what is bioremediation? It is defined as the use of microorganisms or plants to manage the environmental pollution. It is an approach used to treat waste, including wastewater, industrial waste, and solid waste. Bioremediation process is applied to the removal of oil, petrochemical residues, pesticides, or heavy metals from the soil or groundwater. In many cases, bioremediation is less expensive and more sustainable than other physical and chemical methods of remediation. An eco-friendly approach and can deal with lower concentration of contaminants more effectively. Here you can see the circle of bioremediation. First, the delineation of the contaminated sites and the soil sampling is done. Laboratory tests and isolation of microorganisms that break down the pollutants. Excavation of the land is done and the transfer to a place where they will be purified. And construction of the projected crowd, mechanical mixture of the soil, the addition of microorganisms and nutritive substances necessary for efficient degradation of pollutants. And then by reducing the levels of pollutants in the soil, independent institutions carried out the analysis and confirms the results. And restoring the land to the place from which was taken, establishing the old topography. What are strategies for bioremediation in soil and water? Use of indigenous microbial population as indicator species bioremediation process. Bioremediation with the addition of adapted or designed microbial inoculants. Use of plants for bioremediation green technology. What are the strategies? You can see ex situ bioremediation strategy, biopile, bioreactor, composting and land farming. Then biostimulation, bioaugmentation. Consortia, bioaugmentation, recombinant bioaugmentation, biosurfactant, bioaugmentation. In situ bioremediation by natural attenuation, bioventing, biosparging and phytoremediation. Some of the examples of bioremediation technology is phytoremediation. Use of plants to bring about remediation of environmental pollutants. Here you can see the pollutants are degraded or mineralized by the specific enzyme activity. This is called phytodegradation. Pollutants are released into the volatile form through the plant. This is called phytovolatilization. Pollutants are immobilized in the soil. This is called phytostabilization. Pollutants are released through roots. This is called rhizoremediation. Pollutants are degraded by growing roots or microorganisms called phytostimulation. Pollutants are accumulated in harvestable parts of the plant called phytoextraction. What is mycoremediation? Use of fungi to bring about remediation of environmental pollutants. It has biological mycoremediation, chemical mycoremediation and industrial mycoremediation. Biological mycoremediation, many fungi prey and feed on bacteria that are pathogenic to humans. E. coli and Salmonella bacteria are destroyed by mushrooms using natural biofilters placed near the livestock farms and short-line plantings where the runoff occurs. There are as much as 8 miles of mycelium, mushroom filaments, in 1 cubic inch of soil. These make a perfect filter for trapping contaminants from the site runoff. Strains of endangered polypore mushrooms are fighting pox and avian flu viruses. Chemical mycoremediation, natural microbial communities participate with the fungi to break down the contaminants eventually into carbon dioxide and water. Wood degrading fungi are particularly effective in breaking down aromatic pollutants, toxic components of petroleum, herbicides, pesticides. Mushrooms can be trained to break down TNT, PCBs, dioxins and other dangerous toxins. Industrial mycoremediation, living filters can capture hazardous site runoff, chemicals from paper mills, dye manufacturers and power plants. Contaminated brown fields and quarantine mill sites can be bioremediated to break down complex carcinogenic compounds before they leach into the groundwater. So, what is bioventing? A process that increases the oxygen of airflow to accelerate the degradation of environmental pollutants. Here in this image you can see that injection of air or oxygen is done into the soil and there is aerobic degradation of contaminants by the indigenous microorganisms and the contaminated zone is removed by vacuum withdrawal. What is bioleaching? Use of microorganisms in solution to recover metal pollutants from contaminated sites. Here you can see a pyrrhotite with trace of nickel 0.6 to 0.8% is treated with oxygen by the iron oxidizing bacteria, where the ferric is converted to ferrous and the sulfur is obtained, which is a potentially valuable byproduct. Nickel is also obtained, which is a potentially valuable byproduct. The sulfur obtained by this process is oxidized by the sulfur oxidizing bacteria and acid production occurs in the form of sulfuric acid. Bioaugmentation, a addition of selected microbes to speed up the degradation process, where there is cell bioaugmentation, where the pre-adapted bacterial strain or pre-adapted bacterial consortium or genetically engineered microorganisms or commercial inoculum is inserted in the contaminated soil or water matrix and the soil is activated. Gene bioaugmentation, where the bacterial carriers of mobile genetic elements are inserted into the soil and the naked genetic materials are also inserted into the soil to form biofilms and helps in the bioaugmentation. In plant bioaugmentation, the phytoaugmentation occurs and there is rhizoremediation. What is composting? It is a process by which the solid waste is composted by the use of microbes into manure, which acts as a nutrient for the plant growth. We eat lot of fruits and vegetables from healthy plants and some nutrients remain in the food waste or scraps and these are broken down by many worms and microorganisms into good nutrients which are absorbed by the plants and the plants live in a healthy soil. Fruits and vegetables absorb the nutrients from the soil to grow well from the compost. What is rhizofiltration? The uptake of metals or degradation of organic compounds by the rhizosphere microorganisms. Filtering water through a mass of roots to remove the toxic substances or excess nutrients. The pollutants remain adsorbed or absorbed to the roots. The Chernobyl sunflower were grown in radioactively contaminated roots to remove the radioactive contaminants. You can see in an experiment on the right side with a sunflower plant where the contaminated water is inserted into the hydroponic system and the plant roots absorb the contaminants and they are removed by the roots. What is rhizostimulation? Stimulation of plant growth by the rhizosphere by providing better growth condition or reduction in toxic materials. So rhizostimulation, you can see the soils contaminated with heavy metals like PAH, pesticides and hazardous pollutants. The rhizospheric microbes that are present in the soil, they helps in rhizostimulation, and the root exudates, soil pH, carbon dioxide oxidation reduction reactions, secretion of enzymes like IAA, ACC, deaminase, siderophores, biosurfactants and other growth regulators are produced by the rhizospheric microbes, and this helps in the removal or detoxification of the contaminants and improvement of the soil quality and the plant growth promotion. What are the limitations of bioremediation? Biodegradable contaminants can be transformed using bioremediation process. Bioremediation processes must be specifically made in accordance to the conditions at the contaminated site. The use of genetic engineering technology to create genetically modified microorganisms or a consortium of microbes for bioremediation process has a very great potential. Now, what is biofuel, otherwise called algal biofuel? Algal fuel, also known as algal biofuel or algal oil, is an alternative to liquid fossil fuels, the petroleum products. This is also used as a source of energy-rich oils. Also, algal fuels are an alternative to commonly known biofuel sources obtained from corn and sugarcane. The energy crisis and the world food crisis have initiated interest in algal culture, that is farming algae, for making biodiesel and other biofuels on lands unsuitable for agriculture. Botryococcus brownie is used to produce algal biofuel. Here you can see that the algae, which is a renewable resources, by the utilization of sun energy, helps in the extraction of the oil, formation of the protein residues occur, and the biodiesel production occur, resulting in the formation of glycerin products, and the renewable fuel is formed, which is used in the existing technology of transport, and there is also a release of a lot of carbon dioxide. Now, what is biological hydrogen production by algae? It is a method of photobiological water splitting. In normal photosynthesis, the alga Clabidomonas reinhardti releases oxygen. When it is deprived of sulfur, it switches to the production of hydrogen during photosynthesis and the electrons are transported to ferrodoxins. Ion hydrogenase enzymes combine them into the production of hydrogen gas. Here you can see in the image that in the chloroplast of the algae, the PS2 in the presence of manganese, water is taken, helps in the formation of oxygen and hydrogen ions and 4 electrons, and this gets converted by the cytochrome enzyme system and ferridoxin is formed. When the carbon dioxide fixation is available, FNR is used and oxygen is released and carbon dioxide is utilized. Whereas when the carbon dioxide is not available or oxygen is not available, the hydrogen production occurs, resulting in the accumulation of hydrogen. What is bioprospecting? It is the process of discovery and commercialization of new products obtained from biological processes. Bioprospecting may involve biopiracy, in which indigenous knowledge of nature originating with indigenous people is used by others for profit without authorization or compensation to the indigenous people themselves. Bioprospecting has major areas: chemical prospecting like drug and pharmaceuticals, pesticides, cosmetics, food and additives, and other industrially valuable chemical products. Gene prospecting, genetic engineering, crop development, fermentation, and cell culture. Bionic prospecting, like the designs, sensor technologies, architecture, bioengineering, and biomodeling. Now, what is this biopiracy? Biopiracy can be defined as the manipulation of the intellectual property right laws by corporations to gain exclusive control over national genetic resources without giving adequate recognition or remuneration to the original possessors of those resources. So, biopiracy, it has an illegal access. No prior informed consent is obtained and no balanced and fair benefit sharing occurs. This results in patent without or with novelty, other commercial uses, and utilized for research. In legal access, there is potent lacking of novelty and patent infringement occurs as a biopiracy. What are the examples of biopiracy? It includes recent patents granted by the US Patent and Trademarks Office to American companies on turmeric, neem, and most notably basmati rice. All three products are indigenous to the Indo-Pakistan subcontinent. Now, biopiracy of neem. The people of India used neem and its oil in many ways to control fungal and bacterial skin infections. Indians have shared the knowledge of the properties of neem with the entire world. The neem is a miracle tree. Before toothbrushes became common, people in India would chew neem twigs to clean the teeth and the gums. Neem leaves used to treat skin infections, acne, boils, burns, and other skin problems. Neem flowers used in aromatherapy and medicinally to treat anorexia, nausea, and intestinal worms. Neem oil extracted from fruits and seeds used in cosmetics and beauty products such as soaps, hair oil, hand wash, and soap. Pirating this knowledge, the United States Department of Agriculture (USDA) and an American MNC multinational company W.R. Grace in the early 90s sought a patent from the European Patent Office on the method of controlling of diseases on plants by the aid of extracted hydrophobic neem oil. The patenting of the fungicidal and the antibacterial properties of neem was an example of biopiracy, but the traditional knowledge of the Indians was protected in the end. Now, biopiracy of turmeric. The United States Patent and Trademark Office in the year 1995 granted patent to the method of use of turmeric as an antiseptic agent. Turmeric has been used by the Indians as a home remedy for the quick healing of the wounds and also for the purpose of healing rashes. So, the benefits of turmeric: cures acne, heals wounds, cures psoriasis, prevents cancer and growth cells, prevents inflammation, treats diabetes, controls blood pressure. It is used as a face pack, prevents skin damage, exfoliating agent, tightens skin, gives you a glowing skin, lightens the facial hair, reduces cholesterol. So, the journal article published by the Indian Medical Association in the year 1953, wherein this remedy was mentioned. Therefore, in this way, it was proved that the use of turmeric as an antiseptic is not new to the world and is not a new invention, but formed a part of the traditional knowledge of the Indians. The objection in this case, US Patent and Trademark Office, was upheld and traditional knowledge of the Indians was protected. It is another example of biopiracy. Biopiracy of basmati. On September 2nd, 1997, the US Patent and Trademarks Office granted patent on basmati rice lines and grains to the Texas-based company RiceTech. This broad patent gives the company several rights, including exclusive use of the term basmati as well as the proprietary rights on the seeds and grains from any crosses. The patent also covers the process of breeding RiceTech's novel rice lines and the method to determine the cooking properties and starch content of the rice grains. Here, the basmati rice is grown in India, is distinct for its unique flavor and aroma, but an American company got patent rights on basmati through the US Patent and Trademarks Office. The new variety of basmati has been developed by this company by crossing an Indian variety with a semi-dwarf varieties. India has periled the United States to take the matter to the WTO as an infringement of the TRIPS agreement, which could have resulted in major embarrassment for the US. Hence, voluntarily and due to few decisions taken by the US Patent Office, RiceTech had no choice but to lose most of the claims and most importantly, the right to call the rice basmati. In the year 2002, the final decision was taken. RiceTech dropped down 15 claims, resulting in clearing the path of Indian basmati rice exports to the foreign countries. The Patent Office ordered the patent name to be changed to Rice Lines 867. There are 27 varieties of basmati rice grown in India. The patent by RiceTech Incorporation in Alvin, Texas, USA with support of the IRRI. Finally, India won the trial. So today, in Principles and Processes of Biotechnology Part 4, we discussed about the transgenic plants, otherwise called GM crops or genetically modified crops, biofarming, bioremediation, biofuel, bioprospecting, and biopiracy. Thank you. Kindly subscribe, like, share, and comment the channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read Med Prep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.